Hi everyone. Let me just check if I'm audible. Okay, yes. Okay, so structure of a kidney tubule. For a structure of a kidney tubule. Okay, so structure of a kidney tubule. Right, yeah. Okay, so today's topic is structure of a kidney tubule. Okay, so let's have a look at like what all parts are there, the parts of the nephron, what we mentioned. So there is a PCT, Henle's loop, and then DCT. So we will see the arrangement of this in the kidney as well. So before I start, let me introduce myself. I am Vani Kari, biology educator at Unacademy. I am an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. So let's get started, and they join with us in Telegram, so that for homework and class related notes, so you can join the channel Vani Kari ICSE class 9 and 10. So Telegram channel you can click on, so you will find the link in the description box. Okay, so you can go to the description box and find this link for Unacademy ICSE and ISC both link. Okay, and we will be conducting a free special class on Unacademy platform, that is Unacademy app. Okay, so there we will be we will be teaching a class. So there is a real time interaction that is going to occur, so as there is a chat option available, and also there are emoji options are available. So it is a live real time interaction using the chat and emojis. Then you can also ask questions, so you can type in your questions here, or you can attach an image and you can send it to the educator. Again, this is during a live class itself, so educator is going to get a notification and they'll be clearing your doubts. Okay, so other than that, there is a polls which we will be giving. So let's say there will be a question that we will be showing on the slide, and then we will be conducting a poll, so you can choose the correct answer there. So like this we will conduct a poll var. Okay, that means many of the questions will be asked. So as it is your exams are nearby now, so mainly we are conducting a poll var as well as one shot revision for you all. Okay, so you can take the benefit of this one, that is by downloading the Unacademy Learners app. So you will have access to this one. So this is all free and it is a special class. And also we will show you a leaderboard, that is basically to know your competitors. So first, second, third, so you will know your positions. Okay, so this is my special class timetable. So as of now for like Feb 9th, so let's crack biology SQP. So this is sample question paper we are solving. Okay, so this is a sample question paper which we will be solving. So it is for biology, like tomorrow I have, that is on 9th. So 9th I am solving one question paper, and on 13th also the sample question paper I'll be solving. Okay, so you can join me at 5:15 p.m. on Unacademy Learners app, that is platform. So other than that, on Feb 11th I'll be conducting a structure of nephron itself. Okay, so Unacademy subscription features, if you have to look at, so the benefit of this one is you can learn live from the comfort of your home. So unlimited access to all the courses you will have. Top educators of India are going to take the classes for you all in one platform, and there will be a regular doubt clearing session. Okay, so after about every 3 sessions, so we will conduct one session, this 4th session, what is there, so this is a doubt clearing session. So whatever doubts you have related to the topics that are previously covered, so you can clear your doubts in this particular session. Okay, so exhaustive coverage of a syllabus, that means complete syllabus will be covered. Mentorship and guidance is given, study material, the practice test series, live test series, batch courses, So different educators will be having a different batches, and same way different educators are conducting a different courses also. So one is conducting on one particular chapter, the other educator is conducting a course on other particular chapter. So you will have access to all of those courses if you are going for a plus subscription. So there is a daily practice section also, and weekly mock test series also, live quizzes, daily MCQ and subjective test series. So all these benefits you can take from the plus subscription on Unacademy platform, and you can use the referral code VANIYT. Okay, so VANIYT is the referral code where you will get 10% off or 10% discount on the subscription with this referral code. Okay, so you can have a look here. So this is for class 10, 11 and 12. Okay, so for subscription, so if you want to try, you can go for 3 months of subscription, like how exactly this works. And if you are planning to cover this class 11 and 12 also, so maybe you can go for 12 months or 18 months or 24 months of subscription. So based on your convenience, you can go for any of these months of subscription, and this is the referral code VANIYT. Okay, so only if you are using a referral code, you will be getting 10% discount. Okay, so don't forget to use the referral code VANIYT. And other than that, there is a mega offer which is coming up for all the plus subscribers of our ICSE mainly. Okay, so for ICSE class 9, 10, ISC 11, 12. Okay, so for all these classes the offer is applicable. So let's say you are buying a 12 months of subscription or 12 months and more. Okay, so you will get extra like free 2 months of extension. So let's say you are going for 12 months, so you will be getting 14 months of subscription here. So again, flat 10% discount on purchasing a plus subscription of any duration. So this one is valid for first 500 subscribers only. So for this flat 10% off, so all you need to do is use my referral code VANIYT. Okay, so with this referral code only you will get a 10% off. So now moving on to the main part, that is the structure of a kidney tubule. So let's have a look at this one. Okay, so yes, kidneys, when we mention, so these are bean shaped excretory organs. So you know 2 of the organs are there. Okay, so 2 of these organs are there. So there is one kidney on the right side, and then there is another kidney which we will be drawing slightly above. Okay, so this is right kidney, what is there. Okay, so this we always draw slightly below. All right, and the left kidney, when we mention, so this is drawn slightly above than the right one. Okay, so this one is the left kidney now. So these kidneys, what are there? So these are bean shaped excretory organs. Okay, so what do they have now? So they have millions of what? Millions of nephrons will be there in them. Okay, so here million, okay, 1 million over here. Okay, so 1 million over here, so 1 million here, and the 1 million in the other one. So what are 1 million there? So nephrons are, it is not just 1 or 2 nephrons are present. So millions of nephrons, or a kidney tubule, or a uriniferous tubule, or a renal tubule. So different names are there, and don't get confused between the names again, because all these names are the same for the nephron itself. So either we can say uriniferous tubule, or kidney tubule, renal tubule, or a nephron. Okay, so all these names are same itself. So each of these kidneys will have millions of these nephrons. Okay, so now what is the main function of a nephron? It is basically purifying the blood, that is by the process of ultrafiltration. Then reabsorption is occurring, tubular secretion is occurring. So today's class we will have a look at the nephron structure. Okay, so what exactly, how exactly arrangement of this nephron is done in a kidney. So now when we talk of a kidney now, okay, so let's say this is a kidney. Okay, so how exactly arrangement of this nephron is done? So there is a cortex region there. Okay, so outer one is the cortex region which is present. Then this region, what is there? So this is the medulla region. Okay, so this one is medulla. Okay, and this outer region, what you find? So this is the cortex region. When you observe, when you take a longitudinal section, okay, so let's say this is a kidney now. Okay, so this kidney, we are not making a cut here, that is, it will become a transverse section. Longitudinal section means basically we are cutting it from here, so we are cutting it in half from the lengthwise. Okay, so that is the longitudinal section. When the longitudinal section is taken, so cortex region, when we observe, it will give us a dotted appearance like this. Okay, so you will observe dots like this. Okay, so there are dotted appearance that you will find. And in the medulla region, what do you find? So in the medulla region, you will find a striations or a pyramid like appearance. Okay, so I'll just draw this one. So now this is a dotted region. You know, in the medullary region, so you will find these pyramid like structures. Okay, so this is a pyramid like structures what you find now. Okay, so this pyramid like structure will have these lines as striations. So let's see in today's class how exactly this nephron is arranged in here. So we will see that that is in the cortex. So this is not just one nephron which is arranged, millions of nephrons are arranged in a kidney. So we will have a look at that. So now with respect to that, functions of kidney when we mention, so these are excretion and then the osmoregulation. One second. Okay, so functions of kidney is basically excretion, that is to get rid of the unwanted materials. Then osmoregulation, what is this? So it is to maintain the same balance. So ionic balance, the water and the mineral balance has to be maintained. So that is the main function with respect to kidney. So kidneys are gonna help in filtering of a blood. So how exactly blood filtration occurs? So that is by a process called as ultrafiltration, then reabsorption, and also there is tubular secretion which is occurring, mainly the ultrafiltration and then the reabsorption. So they are having a role in maintaining the same concentration of blood by getting rid of the waste products. Okay, so urine is actually the filtrate, that is after the filtration of a blood is done. So you know, let's say I'll explain it here. So let's say there is a sausage that we are taking. Okay, so this one we are filtering over here. Okay, so down here is a vessel. So now this one is a filtrate. Okay, so down which is coming here when we pour a blood over here. Okay, so let's say we are pouring a blood onto this one. So whatever remains here on the top, okay, so that is basically the remains of this blood which is there. So that will be going back, and this filtrate, what is there? Okay, so after certain process, so now this filtration which is occurring through this sausage, okay, so this process we call as a ultrafiltration. And who is acting as a saucer here? It is basically, I mean, sieve. Okay, so sieve it is basically. So who is acting as a sieve here? Sieve is acting. So in case of kidney, it is the you know, there is a structure over here which is called as a nephron. I can show it here. So there is a Bowmans capsule. Okay, so this Bowmans capsule is acting as a sieve. Okay, so Bowmans capsule is acting as a sieve. Okay, I mean it is receiving. So in that, you know, the glomerulus filtration. So Bowmans capsule along with this glomerulus, okay, so they will act as a sieve. So if I have to explain it here clearly, so glomerulus is mainly sieve, okay, that means where the filtration is gonna occur. Then Bowmans capsule, what is there? So it will act as a receiver. So basically it is receiving the filtrate. Okay, so filtrate is going to be received, that is glomerular filtrate, not just the normal filtrate, glomerular filtrate. So let's have a look at this one now. Okay, so here in this case, now this is the process. As you can see, reabsorption, the waste is generated. So we will see the structure first and then you will clearly understand. Okay, so yeah, so kidneys are two bean shaped organs which are about, so you can see this is a cap like structure. So it is the adrenal gland here. So now this is about 10 centimeter long. Each kidney is 10 centimeter long, and you know, so this is about 6 centimeter wide. Okay, so they are 6 centimeter wide. Where they are located on the urinary bladder. So now this is a urinary bladder, so they are located above here. Okay, so that is the location of a kidney. So again your books will ask you a exact location. So in your exam, so you need to write a exact location. So whatever is given in your textbook, so same sentence that you need to write. So that means they are located on the urinary bladder. So now with respect to same, so urine is intermittently emptied from the urinary bladder. So what is happening? So after about it is 2/3 full. Okay, so after the urine formation occurs and after about it is 2/3 full. Okay, so this one, the urine is actually be passed out, that is through the urethra. Okay, so urethra is this one. So this golden color what you find here. So this is actually the urethra. Okay, so how exactly urethra is gonna open? So that is by a sphincter muscle, so guarding. So let's say now this is a vessel over here. Okay, so this vessel, what is there? So this has to open up, right? So now for opening up of this vessel, so there are sphincter muscles. Okay, so this sphincter muscle is gonna regulate the opening and closing with respect to the urinary bladder, so that the through the urethra urine can pass out. So that we call as a micturition. So now here the openings of the ureter, as you can see here, now this is a ureter over here which is opening into the urinary bladder. That means urine is collected from here and that urine is left, or it is reaching the urinary bladder. Okay, so openings of the ureters into the bladder are somewhat projecting. So why do you think that this is projecting? You know, if like whenever the contraction of this urinary bladder, so now one has to urinate, one has to pass their urine. So you know, this urinary bladder muscles, what are there? So they will contract. When the contraction occurs, so there is a chance that the urine which is here, so this can backflow. Okay, so it can backflow back into the ureter itself. So in order to avoid that, so this particular ureter, what is there? So this is somewhat projecting like this. Okay, so when it is projecting, what will happen? So it is the fluid what is there, so it will not easily move into the ureter. So if it is straight, when the contraction occurs, so it can easily move against the gravity, but when it is projecting, the easy movement is not found. Then other than that, they act like a valves to prevent the backflow of a urine also. So backflow of a urine is avoided as they will act like a valves, that is for closing and opening. So there are certain regulatory valves what are there. So those will, it is valves are not there actually, but the projecting ureter itself is acting as a valve here. Okay, then other than that, when the bladder contracts to pass through the urine, now sphincter muscle what is there? So this will guard the opening of the bladder into the urethra. So opening, so use black color what you find here. So this is a sphincter muscle which is shown. Okay, so this sphincter muscle will help in the opening. So if the sphincter muscles will open, then there is a urination or a micturition. If this muscle what is there, so as you can see here, so if this particular muscle what is there, that is a sphincter muscle, so if it is getting closed, then there is no micturition or a urination. So it will guard the opening of the bladder into the urethra and relaxes only at the time of the urination. So sphincter muscle, so let's have a look here. So now the sphincter muscle what is there? So when it relaxes, okay, so when the sphincter muscle relax, then so you can see only at the time of urination, that means urination can occur. That means now this is open. Okay, so sphincter muscle is open so that the urination can occur. So if the sphincter muscle is contracted, okay, so if the sphincter muscle is not relaxed, when it is in a contracted condition, that means it is close, and again there is no urination or there is no micturition. All right, so that is how is it going to open. So let's say relaxation of sphincter muscle will occur, then contraction of sphincter muscle. So it is basically relaxation and contraction is going to occur. So that is mainly from the impulse. Okay, so that is mainly the impulse from the brain is coming, and this impulse is reaching the muscles. You know, the neurons will always act on either the muscles or mainly the glands, okay, and sphincters are basically the muscles. So sphincter muscles are going to receive a signal from a brain, that is impulse from a brain, and based on that either the contraction will occur or the relaxation will occur, okay, and if the relaxation is occurring then it is urination, if the contraction is occurring then there is no urination. Is this clear? And the next one is actually, one second. So internal structure of a kidney, okay, so the longitudinal section of the kidney when we talk, okay, so that is what I was explaining here in this one, okay, so this is the longitudinal section that we will find, okay, so this will show two regions, that is the outer dark one, okay, so it is having a darker appearance when we observe them under a microscope. So there is a darker cortex region, and on the inside there is a lighter structure, so that is a medulla region, okay. So here now this is a kidney, okay, so this kidney when we mention, so on the outside, so this is a longitudinal section that we have taken, so on the outside you will find this region somewhat darker, okay, so this is the darker region, and this darker region is called what? It is actually a cortex region. All right, so this is now a cortex region. Why am I drawing it in a dotted appearance? Because you know, when you study the structure of a nephron, so you if you are knowing if you are having an idea already, there is a PCT, proximal convoluted tubule, the distal convoluted tubule, the Bowmans capsule, so they are all providing this dotted appearance, okay, so dotted appearance of the cortex region, so that is mainly because of PCT, okay, then DCT, and also this Bowmans capsule, okay, so all of these are providing this dotted appearance. Now when we talk of the medulla region, so in the medulla region you will find these pyramids like this, okay, so pyramid structures you will find, so these pyramids will have a striations, okay, so this is now a striation which you will, the lines you will find in here, okay, so there are many lines which are arranged, so these are called as a renal pyramids, okay, so now this whole structure, what is there? So renal pyramid it is called, and this renal pyramid you will find in the medulla region, okay, so this is a renal pyramid now, okay, so this where you find, so this you find in the medulla region. So what exactly does the renal pyramid will have? It is the, so you know, you will have this line like appearance, so that is mainly because of the Henle's loop, okay, so Henle's loop, and also the collecting duct, that is urine which is there, so it is collected into the collecting duct, right, so collecting duct, so both of them together are giving this appearance, that is the striations or line like appearance, so in the medulla region. So now this one is the medulla region, okay, so in medulla region, so you see these lines, right, so lines are basically of Henle's loop and a collecting duct. So how exactly arrangement is done now? So it is basically the cortex is receiving all the PCT, DCT, Bowman's capsule, and the medulla is having the Henle's loop and then the collecting duct, okay. So here with respect to that, the medulla is composed of a finely striped substance which is arranged in several conical pyramids. So these are the conical pyramids, okay. So the apex of each pyramid papilla, so this projects into pelvis. Now you know this is the apex now, okay, so let me just change the color, okay. So now here you know the apex of each of these pyramid, okay, so this is apex is called as a papilla, okay, so this part what is there, so this is apex now, okay, so this is called as papilla, okay, so now this one is actually papilla, and this papilla is projecting where? It is projecting in the pelvis, okay, so now this part what you find here, so where they are projecting, so this is actually forming the pelvis region, okay, so here you know it will continue as a ureter, okay, and this region what you find, so this is a renal pelvis region, okay, so this whole area what you find here, so this is complete completely a pelvis in the medulla region itself, but it is a pelvis because all the papillas are projecting into this one and it is forming a pelvis. Okay, so now this kidney is composed of an enormous number of these tubules, minute tubules. So this is a nephron, a uriniferous tubule, a renal tubule, or a kidney tubule. So all of these are names what are mainly there for the nephron. So they can ask you to draw the structure of nephron, then you need to draw this same one. So uriniferous tubule, renal, or a kidney tubule itself. Okay, so here you can see the structure. Okay, so as you can see here, there is this hilum. Okay, so hilum is basically a junction. Okay, so junction where this pelvis region, so we can just see here in this case. Okay, so okay, I have not added the diagram there. Anyway, I'll just show. So wherever this particular pelvis region is joining with the kidney, okay, so that is this ureter is joining with the kidney, so there we call it as a hilum. Okay, then these are the pyramids and this one is the ureter. This is a pelvis region where the papilla, it is a pelvis, renal pelvis, where the papilla of the renal pyramid are projecting into. Okay, so now this one, as you can see, so this is all papilla. Okay, so this is all papilla now. All right, and then this one is the cortex region which is giving a dotted appearance, and then there is a medulla region. So this center where you find a conical pyramid, so that is actually a medulla region. So it is the same diagram. Okay, so A is a simplified diagram which you can draw easily. So B1 is basically along with the blood supply is given. So the renal vein comes here. One second. Yeah, so there is a renal artery which is coming, that is from the heart, so it is going to both the hearts. Then there is a renal vein which is leaving this one. Okay, so from here, from the same junction, so renal vein is leaving back to the heart, so that is by collecting a deoxygenated blood. Okay, so renal artery is bringing the oxygenated blood, renal vein is leaving the kidney, that is going to the heart, so it will be carrying the deoxygenated blood. Okay, so here the structure of a kidney tubule when we mention, so each kidney tubule is having the following parts. Okay, so Bowmans capsule, so we will just draw this one and then let's see. Okay, so I'll just draw the structure of this Bowmans capsule now. Okay, so here is the diagram. So this is now a Bowmans capsule. Okay, so this is a Bowmans capsule as you can see. So this is somewhat cup-like appearance as you will see. Okay, so now this is a Bowmans capsule. All right, and this Bowmans capsule will continue down here. Okay, so it is forming this convolutions or foldings. Okay, so these foldings you will find. So now these foldings what you find here, so these are called as a proximal convoluted tubule. Okay, so this one PCT. Okay, so this first folding what you find, so this is actually PCT, proximal convoluted tubule. Then this one will extend as such. Okay, so this will continue as such, and there is a formation of a loop here. Okay, so there is a formation of a loop. Okay, and you already know the name for this one, as I already said, it is a loop. So there is only one loop which we always mention. So this is actually Henle's loop. Okay, so this is Henle's loop now. Okay, so there is a PCT, this one is a Henle's loop, and again you will find these foldings or convolutions. Okay, so now this one, as you can see here, so there is another folding which you will find. Okay, so these are joining where, so this is joining the collecting duct later on. So here we will draw the collecting duct. Okay, so there is this collecting duct which is opening again into different formations. Okay, so now this is done. Okay, so this is DCT. So why do we call it as a PCT and DCT? Okay, so yes, I'll explain that. So PCT is proximal convoluted tubule. What is the meaning of this one? when we mention proximal, I'll just write here. So proximal is for nearer. Okay, so proximal is nearer. So nearer to what? Nearer to Bowmans capsule. Okay, so this one I'll just label. So this one, what you find here, so this is Bowmans capsule. Okay, so this is Bowmans capsule. Okay, so as you can see that these tubules, what are there? So this is nearer to the Bowmans capsule, so that is why we mention it as a proximal. Okay, so proximal is nearer to Bowmans capsule. Okay, and then next one is what? So there is a convoluted tubule, so it is a PCT. So next one is DCT. So DCT is basically distal convoluted tubule. So why do we call it as distal? Because this one is far from Bowmans capsule. Okay, so since it is far from the Bowmans capsule, so you can see here the distance is very short here, the distance is far here. Okay, so that is why we call it as a DCT, distal convoluted tubule. And this one, what you find, so this is actually a collecting duct. Okay, so this is a collecting duct. That means what it collects? It collects a urine. Okay, after all the process is done, so urine, once it is formed, so till here the urine formation will occur. Okay, so once it enters here, so this urine is entering, so urine will be leaving through this collecting duct. Okay, so collecting duct is collecting a urine from different nephrons. So here and all there are different nephrons which are connected. So now so I'll just divide this part here now. Okay, so collecting duct we can just draw some more bigger again. Okay, so because it will be larger itself, so we will show some openings also. So this is now a collecting duct here. Okay, so we saw the cortex region and then the medulla region, right? So this part, what you find here until here. Okay, so this part is forming the cortex region. So I'll write it here itself or on this side. Okay, so I'll write it here. So this region, what you find, so this is actually the cortex region. So in cortex region, what all comes? DCT, PCT, Bowmans capsule. And this region, what you find here, so this is actually the medulla region. Okay, so in medulla region, what do you find? So medulla region, you find the Henle's loop and then the collecting duct. Okay, so this is forming the medulla region. All right, and this is forming the cortex region. Okay, so now moving on to next one. So here, one second. So there are arterioles which are coming. So we saw here in this one, I'll just explain now. Okay, so let's draw a kidney again. So this is a kidney here. Okay, there is renal artery which is coming. Okay, so that is from the heart, a renal artery is coming. Okay, so you know that artery has to divide into arteriole, and how many nephrons are there? Millions of nephrons we can find in the kidney, and to all of these millions of nephrons, arteriole has to be supplied. That means once it enters the kidney, so this will divide a million times. Okay, so renal artery will divide, branch, rebranching will occur. So that means it is forming this particular arteriole. Okay, so there is a formation of arteriole which we can see. Okay, so renal artery is dividing million times to form a arteriole. So this will be going where? It is going to the nephron itself. So let's say this renal artery. Okay, I'll just draw it here. So renal artery enter the kidney. Okay, so it has divided into arteriole now. Okay, so now this is a arteriole which is divided into two of the parts. So let's say I'm just drawing two parts here just for the understanding. Okay, so this was a artery, the bigger one, and this what you see, so this is arteriole. Okay, so this is arteriole. Now these have to enter where? So this will be entering the Bowmans capsule. So let's draw here. So now this is gonna enter here. Okay, so this is gonna enter into this structure. All right, so that is the Bowmans capsule. So we will just draw it like this. Okay, so this is leaving again. So now since it is entering, so we call this as a afferent arteriole. Okay, so this is called as a afferent arteriole because it is entering where? It is entering the Bowmans capsule. So what will happen? So there will be formation of these capillary. It is dividing further and further and it is forming the capillary, and you always need to make a note here. So I'll explain about this one. So it will divide further here. Okay, so many divisions will occur. So it is forming a bunch of capillary here. Okay, so now this is a capillary which is formed. Okay, so this is a afferent arteriole, and the one which is leaving here, so this is called as a efferent. Efferent is leaving. Okay, afferent is coming. So coming where? It is coming, or it is getting into the Bowmans capsule. Efferent, when we mention, it is leaving the capsule. Okay, so now here in this case, so now this is the efferent arteriole now. Okay, so this efferent arteriole, what is that? So this is leaving the Bowmans capsule, and this capillary structure, what you find, so this is called as a glomerulus. Okay, so this is a glomerulus. Okay, so glomerulus is nothing but a capillary itself. Okay. 1 second. So now this glomerulus and together with the Bowmans capsule, so both of these together, so we have named them a different naming. So we have given it a different name. So that is nothing but, so both of these together we call it as a Malpighian capsule. Okay, so this one is Malpighian capsule. Okay, so Malpighian capsule, when you hear the term, it is basically including the Bowmans capsule and a glomerulus. So both of them together it will form a Malpighian capsule. So this whole structure what you find here. Okay, so this one, so this whole structure what you find here, so this is called as a Malpighian capsule. Okay, that is Bowmans capsule with the glomerulus. Now what is going to happen? So this is the afferent arteriole here. Okay, so this one is a capillary and here is the efferent arteriole. Okay, so now these have to divide further. So this is now dividing further where it will be surrounding. So now this is a capillary. Okay, arteriole is there. So again it will form the secondary set of capillary. So let's have a look at this one now, and always make sure that while you are drawing, the diameter of this one is more as compared to the diameter of this one, because as the diameter is decreased here in the afferent arteriole, that means the volume is decreased, right? So you know the law, right? So basically when the volume increases, okay, so volume increases, or here we are talking about the volume decrease. Okay, so in the afferent arteriole what is happening? The volume, the diameter is decreased. Okay, so volume decreases, so what is happening? The pressure is increasing. So what is happening? So higher pressure, so higher hydrostatic pressure is developed. Okay, so pressure is basically a hydrostatic pressure here. Okay, so that is mainly with respect to the liquid which is filtering, which is getting filtered. So here in this case now, so as there is a decrease in volume, so what is going to happen? The blood which will flow through this one. Okay, so the blood which has to flow through this one, I'll just change the color. Okay, so now here in this case what has to happen? The blood has to flow through this. Okay, so the blood which has to flow through this capillary and the filtration which has to occur, so the blood which is leaving the afferent arteriole, so that will be a thicker blood. Why it is a thicker blood? Because all the liquid part of a blood which was there, so all the liquid part of a blood got filtered out here. Okay, and that we call as the ultrafiltration process, as the blood was flowing with the pressure, and you know here also the volume decreases there. So whenever volume is decreased, the pressure is higher. So what is happening? Because of the higher pressure which is created over here, so you know the liquid part of a blood will all get filtered here into the Bowmans capsule. So what is Bowmans capsule receiving? The Bowmans capsule is actually receiving the glomerular filtrate. Okay, I'll write where I'll write it here. So here it is received is glomerular filtrate. Okay, so glomerular filtrate will be received in here. Okay, so now here you know, so now this structure, what is there? This afferent arteriole. So this is further dividing and it is forming a capillary. So we will just draw that. Okay, so now this one further divide as such. Okay, so this will come here. So this will be further dividing, which will be surrounding this PCT. Okay, so this is gonna surround the PCT, so there will be capillary. So secondary set of capillary formation we can find in here. Okay, so it is further dividing as such, and you know again here they'll join, and again there will be a further division which we can see. So this is again further dividing in this. Okay, so capillary formation will occur. So any idea why exactly capillary formation occurs here? So this is mainly for the reabsorption process. Okay, so now this afferent arteriole, so it will further divide around the PCT, it will further divide around the Henle's loop, and again all of these will join together. Okay, and again there will be a further division which we can see around the DCT also. Okay, so around the DCT we can find a further division. Okay, so as you can see here, okay, so these are all going to join later on, and this is gonna join where? So this will be joining the renal vein. Okay, so this is gonna join the renal vein. Okay, so it was coming from a renal artery. Okay, so it was a renal artery here. Okay, where it divided into arteriole. Okay, then what happened? The capillary formation was done, and now this one is a venule actually. So this is going to join a renal vein because same concentration of a blood has been maintained. And you know, so this Henle's loop, what you find, so this limb, what you find, the one which is coming down, so this we call as a descending limb. Okay, so this is a descending Henle's loop. Okay, and then there is the ascending Henle's loop. So this one is going up, so this is moving in this direction. Okay, then there is the movement in this direction. So here it is a ascending limb or a ascending loop of a Henle. Okay, so this is a ascending limb, and these are the structures. So let's before I continue, so let's just read this part. So Bowmans capsule, when we mention, so it is a thin walled, a single cell thick epithelium. One second. Okay, so you know this is a Bowmans capsule now. Okay, so it will be having a thin wall, so there is a opening here. So this is single walled, that means only a single cells will be there. Okay, so single, only, there is no multiple layers of a cell. So now this is one layer, there is another layer like this, another layer. So like this multiple layers of cells are not there, only the single cell thickness. So now this is one cell thickness here, here also you will find one cell thickness. Okay, so on the both the side you will find one cell thickness. Okay, so single cell thick epithelium, so this is now a epithelial cell it is. Okay, so here single cells what you find, so these are all epithelial cells. Okay, and you know as you can see it is a cup shaped one, something like a hollow ball. Okay, so hollow ball is gonna fit deep inside into this one, so press deep on one side, so its hollow internal space continues into the tubule. So this hollow space, what is there? So this will continue into a tubule like this. Okay, then what is happening? So outer concavity of the cup lodges a knot like mass of blood. Okay, so knot like mass of blood is lodged where? So you can see here the outer concavity is basically a space, okay, where a knot like, so knot like formation you can see in here. Okay, so this is basically because of the afferent arteriole and the efferent arteriole which are living. So further division of capillary will occur. So it is like a knot like mass you can find, which is mainly of a capillary. So this is actually a capillary, a blood capillary itself, which we call as a glomerulus. Okay, so this is called as a glomerulus now. Okay, so now here the Bowmans capsule and the glomerulus. So this one is a Bowmans capsule and then this is a glomerulus. So both of them together we call them as a Malpighian capsule. Okay, or just a renal capsule also. So first one is proximal convoluted tubule. So this we saw. Okay, so proximal convoluted tubule is basically the convolution, the first one. So this is lying next. So proximal means nearer to the Bowmans capsule. Okay, so both the Bowmans capsule and the proximal convoluted tubule, so they'll lie. Okay, so it is just that here they have not mentioned, but the distal convoluted tubule also is lying in the cortex region itself, which is providing a dotted appearance in a sectional view. So this is the structure of this one. So these are, this efferent arteriole will further divide, here it will form a capillary, again here it will further divide, here it will form a capillary, again here it will further divide, again it is forming the capillary, and then later it is joining the renal vein. So same structure I have drawn here. So that is the structure with respect to the nephron and then the capillary network. So loop of Henle, descending limb, ascending limb, the capillary network here, afferent arteriole which is wider, the efferent arteriole, so this is narrower, the diameter is less there. Then there is a glomerulus, a knot like mass of capillary. So proximal convoluted tubule also can be called as the first convoluted tubule. Then there is a second convoluted tubule which is called as a distal convoluted tubule. Then there is a collecting duct where the urine formation, so once the urine formation is done, so this urine is going to enter in here, the collecting duct is going to receive this urine. Okay, so other than that, so we are done. So this is with respect to functioning. Okay, so here anyway the functioning, so today's class was only on the structure of a kidney tubule. So functioning of this one we can discuss, I'll be conducting a special class also on Unacademy platform. Alright, so we will have a look at the functioning also. So you can download the Unacademy Learners app and join in there. Alright, so now with respect to this, another name for the renal tubules is, so neuron, uriniferous tubule, kidney and renal. So yes, anybody knows the answer for this one? I don't see a chat here. One second. Anyway, I don't see the chat. Alright, so this one is actually the uriniferous tubule. Okay, so next question is Bowmans capsule and glomerulus. So together we call as what? So it is Malpighian capsule, the filtrate, renal tubule and then the kidney tubule. Okay, so yes, for answer for this one is again a Malpighian capsule itself. Okay, so next is the reuniting capillaries of the glomerulus form. So there is the afferent arteriole which is coming, then the capillaries will form, that is division will occur, then all of them will unite together and they'll form a efferent one. Okay, so reuniting capillaries of glomerulus are called as a efferent arteriole. Okay, so opening of the sphincter muscles for micturition, that is urination, so it is by the impulse from brain, whether it is true or false. So this one again it is true. Okay, so before I end the class, let me just say about this one. So Unacademy presents a prodigy for class 7 to 12 learners, where it is gonna check your mental ability. So you can enroll now for free. So you can also win scholarships and prizes while testing your knowledge in mental ability. So it is basically a aptitude test. Okay, so what you can do is you can enroll for this one So there is still a time is remaining. So on 13th of Feb you can take up this test at 12 p.m. okay. So this is at 12 p.m. on 13th of Feb. So all you need to do is download the Unacademy learners app. Okay, so this is on 13th of Feb now, because all these are already done. Okay, so for class 9 to 10, so you can take up the test at 12 noon. All right, and you can win exciting rewards in every test. So you can, so rank 1 is gonna get the MacBook Air, the rank 2 and 3 are gonna get Apple iPhone, rank 4 and 5 will get Apple iPad, rank 6 to 40 will get Amazon Echo Dot, then there is rank 41 to 100, so they'll get Amazon watches. Okay, so scholarship slab for others is basically, so rank 1 to 10 will get 100% scholarship slab, then rank 11 to 25, so they'll get 75%, 26 to 100 they'll get 100% 50% scholarship, then rank 101 to 200, so they are getting 25% scholarship, rank 200 to 500, so they are getting 15% scholarship with respect to the same. Okay, so win a 20 lakh college grant. Okay, so this is the test on 29th of January as well as 13th of February. So this 13th of Feb is nearby, so you all can take up this one. Okay, so now these are ongoing batches on Unacademy. So there is a Udan batch, the mode of delivery is Hindi here, and then there is a Champions batch, the mode of delivery is English here. Okay, so all the subjects will be covered, and you know some classes are already done, so you can watch the recordings as well. Okay, so we are also discussing the sample question papers. So I am conducting a class on, taking a special class for SQPs, that is specimen question papers, on 9th and 13th. So that is at 5:15 p.m. Okay, so you all can take up this session. So we will solve some questions from your specimen question paper. Okay, so other than that, so this is the same. So this is with respect to biology I told. Okay, so all other teachers who are there, the educators who are there, so physics will be taken by Kalyan sir, chemistry by Manisha ma'am, maths by Shivam sir, and as well as bio by myself. Okay, so also we are conducting a one-shot revision, so that is for class 9, 10, 11 and 12. So this is again started. Okay, so here I'll be solving the important questions for your final exams for class 10 and 9 as well. So to unlock, you can use the code VKIC10. So it will ask you a code, so you can use that code. And other than that, so ICSE class 10 Ace Your Race. So here Kalyan sir and myself again on 13th of Feb 11 a.m. Okay, so we are discussing about, so we will be helping you about the preparation strategies for your exam. Okay, so we will have a look at the preparation strategy for your exam, and time management during and before your exam we will have a look at. So this is with respect to time management also. Then we will discuss about the answer writing hacks. Okay, so we will discuss all this in the Unacademy special classes. So you will have access to this one. So all you need to do is download the Unacademy Learners app now and try all of these features for free. Okay, so how do you exactly watch these special classes? So all you need to do is download the app, click on get started, then you have to enter your mobile number and a OTP, then you can sign up using your email ID, then you can choose your class. So that is class 6 to 12 you can choose. Then you can choose your ICSE category, then choose the language of your preference. So English, Hindi, English, you can choose this one, and you know you can subscribe for this channel also. So here once you do that, so you will have access, so you can take up all the sessions which are going on, the classes which are going on. So there are free live classes, there are quizzes, Okay, so you can take up all those from the learners app. So download the learners app, and if you are planning on going for a subscription, so you can use my referral code VANIYT to get 10% discount on your subscription. Okay, so you can also find, so use my code to unlock the free courses that are there. So there are many free courses which are available on Unacademy platform, so you can use those free courses as well. All right, that is to unlock them and use it well. Okay, so subscribe on YouTube. Okay, and also you can download the Unacademy learners app and join in there for the special class as well. Okay, so see you all in the next session. So next session I'll be conducting a SQP on Unacademy platform, so you can come join that. So see you all. Bye bye. Take care everyone. Thank you so much.